The overall goal of this procedure is to isolate, expand, and differentiate mesenchymal stem cells derived from canine ovarian tissue.
Hi, I am Dr. Garcia, a professor at São Paulo State University, Brazil. My PhD student, Amanda, will be showing you the procedure today.
Hi, my name is Amanda Hill, and I'm the group leader at the Stem Cell Lab in the Animal Reproduction Department at São Paulo State University. Today, I will be demonstrating for you the isolation and differentiation procedure of mesenchymal stem cells, MSCs, from the ovarian tissue.
This experiment was performed with the ovaries of 4 mongrel female dogs donated after elective surgery at a canine sterilization program. This experiment was approved by the Ethics Committee on the Use of Animals of São Paulo State University, FCAV. The main steps of the isolation are summarized here schematically. Step 1. After castration, transfer the ovaries into sterile tubes with PBS on ice. Step 2. In order to wash the ovaries, they should be transferred into a 100 mm plate with 10 milliliters of PBS. Step 3. Mince the tissue into small pieces, approximately 1 millimeter in size. Step 4. In order to liberate the cells from the tissue, transfer the pieces of